All right. Good afternoon, everyone. You prepared for a good time? Yeah. All right. Thank you. You've all given me a very good time, so thank you very much. I'm prepared to have more fun. So the topic for the discussion this afternoon, I'm just going to, there are, actually let me offer a few thoughts. There are so many possible directions that we could go in with this conversation, talking about different insects, talking about specific pests that and management strategies for those pests. So I'm going to dive straight into some of the background principles and the thinking of how we think about disease and insect resistance. And then if at any point in the conversation, I'm going to probably go into fire hose mode here in a little bit. But if you have any questions for me, please put your hand in the air. I'm happy to take any questions. We have a roving mic if there are any questions. And I certainly want to incorporate any questions that you have and get those answered. All right, so the foundational principle, the foundational premise, and the idea that that I'm approaching this conversation with is the idea that diseases and insects do not occur at random, that they show up in specific places in response to specific environmental stimulus and cues. This idea was first captured and communicated well in the 1940s by William Albrecht when he said that insects are nature's garbage collectors and diseases are her cleanup crew. We all know from our experiences that diseases and insects don't show up indiscriminately, that we will often have certain sections or parts of a field that will show pressure first. And the question is why? What was it about that particular space or that particular environment that created the conditions for that pest to become a problem? You know, when we think about disease susceptibility, disease resistance, I would suggest we need to think differently about this concept that is known as the disease triangle. So the disease triangle has been discussed and considered at length in IPM, Integrated Pest Management, in North America, and I would assume here as well. And the foundational idea is that there are three pieces that are required, completely essential, in order for a disease to express itself. The first is a susceptible host. And the second is a capable pathogen. And the third is an optimal environment, an optimal climate. And the environment for different organisms is considered as being the optimal humidity levels, temperature thresholds, etc. But I would suggest that we haven't given enough consideration to what the definition is of a susceptible host. Because historically, we have the common framework is that any living plant, let's say if we're talking about a wheat plant or rapeseed, that just the presence of the plant itself means that it is inherently susceptible. But that's a false idea, because the immune function of plants can range dramatically. We can observe from actual field practice and experience that some are much more susceptible than others. How many of you have heard of the research work of Philip Callahan? Okay, a handful of you have. It's fascinating. Philip actually did a great deal of his research and had a very close connection to Ireland 50 years ago. Philip Callahan was an entomologist. He was a radio signal RAF operator during the Second World War in Ireland, and he took those experiences following the war. He started studying insect communication systems, and a great deal of his research was classified for several decades because it was used to develop radar cloaking devices, anti-radar cloaking devices for military aircraft. And what Philip identified is that all insects are communicating in the electromagnetic spectrum. They are constantly communicating with their environment, they're communicating with plants, and that in particular, the majority of the insects that we deal with in an agricultural setting, an agricultural context, are communicating with their environment in the infrared spectrum. And the foundational essence, the summary of his decades of research, was that when we have healthy plants out in the field, plants that have a functional immune system, they are photosynthesizing very well, They have a very nice dark green color. When we look at those plants, this is what we see, and this is what insects see. Nothing. They don't see it at all. It appears completely invisible to them because the plants have a very similar infrared signature as the rest of the background environment, the rest of the background soil environment, etc. If, however, we have plants where we have the presence of insects that have imbalanced nutritional profiles, and I'll speak in just a bit about what these imbalanced nutritional profiles are. When we have imbalanced nutritional profiles, this is what we see, and this is what insects see. And I had the fun of finding the image of this sign, but this is a direct quote from one of his articles. He described that in the infrared spectrum, insects perceive imbalanced plants as a neon light against a dark background. That is the calling signal. And the distance this signal can travel is determined partially by humidity and partially by air temperatures. But he said that this signal can be perceived by many agricultural insects for a distance of 700 to 1,400 meters. So significant distance. What this means, of course, is that if we apply an insecticide and we reduce the insect population, we actually have done nothing to solve the problem. The signal's still there. The sign is still there. And if we want to prevent these problems systemically from their very foundation, we have to address the root causes and we have to remove the sign. So this image is the bottom side of a tomato leaf at 26,000 times magnification. No, sorry, that's not correct. I forget what the magnification is here. Maybe 400x, even that seems high. But this is the bottom side of a tomato leaf, covered with trichomes. These small antennae of the plant are constantly communicating with their environment. The plant is constantly communicating with the environment in the electromagnetic spectrum. These small antennae, these small trichomes are also photon receptors, and they're vibrating in the 26 to 36,000 megahertz range. That's where the 26,000 number popped into my mind. So this is the plant communicating with the environment in the electromagnetic spectrum. Here we have a moth antenna, and there is this beautiful fractal geometry that occurs on moth antenna. Oops, did I miss this? So the only difference between this antenna, well, there are two differences between these two antennae. This antenna is about an order of magnitude more sensitive and more accurate. It has a remarkable receptor capacity and is very sensitive to signals in the environment. But the other major difference between the two is the wavelengths that it picks up. But not only are the wavelengths harmonics of the antenna length, but there is also this beautiful fractal geometry component where you take each individual strand of moth antenna and you put that underneath a microscope, and this is what that looks like. And now you take each of the smallest hair that you can see in this image and you amplify that, and this is what that looks like. Isn't that incredible? Insects are communicating in the electromagnetic spectrum in much broader ranges even than what were originally identified in Philip Callahan's work as a reflection of these different antenna lengths. So I wanted to describe this research as to provide kind of a foundational perspective that these insects and these, I've come to really dislike the word pests because pests implies a negative relationship. When in reality, when you back up and you look at this from a broader ecosystem perspective, insects show up to consume and feed on plants that are not fit for human consumption or for livestock consumption. That's really what they are there for. I've frequently heard the comment, particularly applied to organic fruits and vegetables, that, oh, my organic unsprayed apples are fit for the worms to eat, therefore they're good enough for me to eat. That is completely wrong. The fact that they have insect pressure, the fact that there are worms feeding the fruit means that they are not fit for human consumption. They are not in an optimal state of health. They do not have a functional immune system. So yesterday, in my initial presentation, I provided a rapid overview of the plant health pyramid. I'm going to, again, give you a rapid overview, but in slightly more detail. Let me see what I have here. Yeah. All right. Let's just bounce through these. So for those of you who missed the discussion yesterday morning, the plant health pyramid came about as a result of, as we worked with hundreds of growers in many different climates and environments and ecosystems, work with a broad diversity of different crop types. Even across these many different crop types, many different types of plants and different pests, we observed this very similar development of plant health and resistance to different groups of diseases and different groups of insects based on what was happening with plant physiology and what was happening with a plant's nutritional profile. So I'm going to go through these, each of these 4 sections in a bit of detail, and want to give you some practical perspectives on how to manage and how to achieve each of these outcomes. So the foundational level of the plant health pyramid is achieving complete photosynthesis. And what I'm describing here is when plants have the proper nutritional balance, they will move from a foundational threshold of 15 to 20% photosynthetic efficiency to a higher threshold, preferably greater than 30%, and optimally upwards of as high as 60% photosynthetic efficiency. And when this happens, we get 2 things happening. First of all, the volume of photosynthesis increases in each 24-hour photo period, where you can have 50% to 4x greater sugar content every day, actually as much as 6x greater depending on the specific context. So you get a larger quantity of sugars that is produced in each 24-hour photo period. And this is such an important piece. There's a whole separate presentation that I don't really have the time to get into, but what we've observed is that we can actually regenerate soil health and rebuild soils very quickly while we are growing a crop once we have higher levels of photosynthetic efficiency, because even though the plant might produce 2 or 3 times higher sugar content, it's not necessarily going to produce 2 or 3 times higher yields. All of the surplus sugar that has been produced, the majority of it goes out through the root system as root exudates to feed soil biology, and that is how you sequester carbon and build soil organic matter very quickly while you are growing a crop. So 2 things happen here. First of all, the volume, overall volume increases. But the second piece that occurs is that the carbohydrate composition changes. The ratios of reducing to non-reducing sugars, the proportions of complex carbohydrates, the percentage of cellulose and hemicellulose and lignin and so forth changes within the plant profile. And I'm actually going to preempt a bit one of the stages when we get to, let me see, I'll come back to this. This actually, at this particular stage of plant health, we observe, so we have increasing volume, and at this stage, plants become resistant to all of these soil-borne fungal pathogens. So Rhizoctonia, Pythium, Fusarium, Verticillium, etc. Then, so the question is, what do you have to do to achieve this? This is the foundational level. This is the first piece that needs to happen. And plants need to have adequate levels of these 5 elements to get to this stage. And I think I have enough time. I'm going to give you a quick synopsis of the rationale and the reason for being for each of these elements. So I want to be very clear. I'm not suggesting that you need to add these 5 elements to a crop. I'm only suggesting that you need to make sure that the plants have enough. They may have generous levels of 3 out of the 5 or 4 out of the 5 and be completely deficient in 2. And that is going to, those 2 will be the limiting factors, the bottlenecks that limit overall photosynthetic efficiency. So I'll give you a very quick overview of why these are important. So 3 of these elements are foundational to building chlorophyll. When you think about increasing photosynthesis, there are several mechanisms that contribute to increasing photosynthesis, but one of those mechanisms is that you can increase the concentrations of chlorophyll within the leaf. And the levels of chlorophyll within the leaf are a reflection of the supply of 3 elements: nitrogen, magnesium, and iron. When I ask the question at conferences and presentations, I frequently ask people the question of, how do you turn a plant dark green? And it's fascinating that I get completely different answers depending on who and where I'm talking to. When I have this conversation with an agricultural audience, such as here, the answer that comes back is almost universally nitrogen. We put on nitrogen, we turn a crop dark green. When we speak with greenhouse growers, and we ask them, how do we turn plants dark green? The answer is magnesium. They're growing petunias and flowers in a greenhouse environment. When we speak with turf producers who are managing golf courses or growing turf, the answer is iron. They're using 3 different tools to accomplish the exact same thing, but as a result of not using a balanced approach, they create imbalances and they create a suppression of plant health. The optimal approach from a health and photosynthetic efficiency perspective is to use a balance of all 3. So nitrogen, magnesium, and iron. And by the way, I had the privilege of visiting a farm on Tuesday, and so I have a case study of exactly 1, not enough to have an informed and qualified opinion. But my perception from that particular operation was that this particular operation had calcareous soil, limestone-based soil, and it's my understanding that many of you have similar soil types. And in this limestone-based soil, I walked into the field, and it was immediately, like, I, of course, have lots of experience and the benefit of understanding what it is I'm looking for. I could see manganese and iron and magnesium deficiencies from a distance. It was very painfully transparent and painfully obvious. These calcareous and limestone-based soils are going to limit the supply of available magnesium and manganese and iron. And I'll get into each of these in a bit more. So magnesium is the center atom, the center ion of the chlorophyll molecule. The chlorophyll molecule is structured around. It's surrounded by 4 nitrogens. This is why both magnesium and nitrogen will increase chlorophyll concentrations and make a plant dark green. Iron does not directly participate in chlorophyll structure, but you could think of iron as the assembly wrench. Iron is required to build chlorophyll and to put chlorophyll together. And iron is also the foundational enzyme cofactor for many of the carotenoids in plants. And really, when you have, actually this is also worth mentioning, so chlorophyll itself actually intercepts only a very small part of the visible light spectrum. But then there is this group of carotenoids that are all iron-based compounds that intercept a much wider array of the light spectrum. So when you have plants that have abundant levels of iron and high concentrations of carotenoids, they actually intercept a much broader array of light. And again, this contributes to increased photosynthetic efficiency. And by the way, in North America, where I have lots of data to back this up, and I would suspect or expect the same to be true here, crops are almost universally deficient in iron because of soils being very oxidized. And from what I've observed so far, the same is true here, and I'll explain that more once I've talked about manganese. Manganese importance in this process can hardly be overstated. Similar to iron, crops are almost universally deficient in manganese today for a combination of reasons. First, let's talk about what manganese does inside the plant and why it's important. During the photosynthesis process, plants absorb water from the soil, they move it up into the leaves, and the first interaction, the first step with the water molecule is that the water molecule needs to be split in this process called water hydrolysis into hydrogen and hydroxyl, H and OH. And this process of water hydrolysis is facilitated by an enzyme that requires the enzyme cofactor of manganese. This means that when you have a plant that appears visually perfect and conditions appear to be perfect, you have perfect temperature, lighting, abundant levels of carbon dioxide, abundant chlorophyll in the leaf. When that plant doesn't have enough manganese, manganese becomes the bottleneck and the limiting factor in this water hydrolysis process that prevents plants from fully photosynthesizing. So phosphorus, I'll just touch on briefly. Phosphorus is not directly required for the photosynthesis process. I'm sure all of you are familiar with the visual symptoms of a phosphorus deficiency when you have plants that discolor and produce purple veins or purple leaf edges as a result of not having enough phosphorus. What is actually happening in those plants is because there isn't enough phosphorus present to form ATP to metabolize sugars, the plant instead converts those sugars to anthocyanins. And so you get this dark purple coloration. The anthocyanins are the same compounds that turn berries purple and red. They're actually medicinal compounds. But what happens, we are used to seeing this effect in cool soil conditions when soils don't release phosphorus very well. But can you imagine seeing that effect in the middle of the summer? There's one pathway to producing that effect in the middle of the summer, and that is to put on a foliar application, let's just say of magnesium, iron, manganese, and nitrogen, and you facilitate a spike where photosynthesis goes from 15% up to 60% photosynthetic efficiency, and all of a sudden the plant has 4 times more sugars to deal with and to metabolize than it did yesterday. And if there's not enough phosphorus present, you can actually produce this purpling of the leaf from the anthocyanin accumulation in the middle of the summer. It's a lot of fun when that happens, but people get really freaked out. So at this point, I'd like to speak a bit more about iron and manganese and why they are almost universally deficient and what this means from an agronomic context. Again, this is a nuanced conversation, and it's I'm very cognizant of addressing a number of different pieces, but the trace mineral metals of iron and manganese and copper and cobalt, those 4 elements in particular, are required, the plant is required to absorb those and can only utilize those when they are absorbed in what is called the reduced form or the reduced oxidation state. So you sometimes see these elements written as with a double positive sign or a triple positive sign. Those reflect reduced versus oxidized oxidation states. So without getting bogged down in the biochemistry, I want to just give you a very practical example. If you take a piece of iron or steel and you expose it to the elements, to air, sunshine, water, it begins rusting. That rust is oxidized iron. And it is oxidized iron along with oxidized manganese and oxidized copper and oxidized cobalt that are predominant in our soil systems. And plants cannot absorb them and utilize them effectively. So it is very common phenomena for us to observe soil analysis that have extremely high levels of iron, and yet the crop is functionally deficient in iron. And it's very easy to prove or disprove this. Simply get a high-quality iron chelate that is in the proper form, in the reduced form, and put on a foliar application, and you can write your name on the lawn or do whatever fancy curlicues you like, and you will see that signal usually in 24 to 48 hours. Plants turn dark green very quickly. That is proof that even though your soil analysis shows that you have excessive levels of iron, the plant itself is actually functionally deficient. And the same is true of manganese. Now, if you want to dig into this topic deeper, I have a couple of webinars and an online course on redox and reduction oxidation reactions. The challenge is that today our soil microbial populations have become so degraded. It is the function of a healthy soil microbial population to convert oxidized iron and oxidized manganese into the bioavailable reduced form. But in order to do that, soils need a reducing environment, and you actually benefit. You have some of this in ways that many growers in the States don't, and in other places, many other places around the world, because one of the ways of producing a reducing environment is to have a saturated soil. And you have saturated soils for some months of the year, which produces all types of other problems if it's overdone, when you have excessive saturation or for too long a period. However, reduction versus oxidation is a spectrum, just like pH is a spectrum. All of you are familiar with pH, a spectrum from 0 to 14, a neutral point of 7. Redox is also a spectrum from extremely oxidized to extremely reduced. Oxidation is not necessarily inherently a bad thing. But the problem is that our agricultural systems today are skewed all the way to the end of the spectrum of being extremely oxidized. Imagine if we managed our soils to constantly have alkaline pHs, pHs of 10 and 11 and 12. What happens is you gum up the system, you plug up the system, you no longer have good nutrient availability at those high pHs, and the exact same thing is happening when we have these highly oxidized soils. So some examples of things that we do that have a very strong oxidizing effect is when you till soil and you introduce oxygen, that has an oxidizing effect. When you have bare soil that's exposed to the sunlight, that produces an oxidizing effect. When you add limestone, limestone has an oxidizing effect, but not nearly to the degree as soluble ion fertilizers. If you add salt fertilizers that have a high electrical conductivity, that also has an oxidizing effect. In short, if you have a fertilizer that would cause a burn or burning sensation on an open wound on your finger, that burning effect is oxidation of cellular cell membranes. The same thing happens to the soil microbial profile. All of these materials have an oxidizing effect, and that is a pretty good description of our modern farming practices. Oh, but I've missed the biggest one of all. The biggest one is glyphosate applications. 10 ounces of glyphosate per acre are enough to completely shut down the specific microbial populations that are responsible for converting oxidized manganese and iron back into the bioavailable reduced form. All right, enough about that. As you might have figured out, these pieces, magnesium and iron and manganese, are almost universal in our recommendations when we're working with growers. We see that those three pieces are foundational that we need to address. Nitrogen is frequently over-applied to compensate for the sins of these other elements not being an adequate supply. Yeah, go ahead.
Sorry, I'm just picking you up on something you said yesterday, that your approach is not to sort of take people off a chemical treadmill, if you like, straight off, but you've just now said that glyphosate is very disruptive to a healthy photosynthetic system. How do you balance that with what you said yesterday?
Well, the way that you balance that, there are several pathways. First, if it is necessary, well, I don't like that word. If we feel the need to continue using glyphosate, then we have to ask ourselves the question, how do we balance that out, and how do we build and develop a microbial population that can rapidly metabolize the glyphosate in the soil profile and prevent it from building up residues over time? So we have to balance that equation. If we're adding glyphosate, how do we compensate for that? But secondly, I don't particularly like framing the conversation in terms of it's either glyphosate or no glyphosate. There's a broad spectrum. I'm less familiar with the regulatory environment here, but there's a broad spectrum of other herbicide and other weed control options that we need to consider, and perhaps we need to change the overall cropping system as well, so that we no longer need to utilize that. If I have time, I'd like to also speak a little bit about weeds and how we can use nutrition management to change weed pressure. We can change weed pressure significantly with nutrition management of the soil profile. All right. Next stage, complete protein synthesis. So what is happening here at this stage is when we have plants that plants are absorbing nitrogen from the soil in its various forms. I'll talk about this in a bit more in my next presentation, but you have 4 major different forms that can be absorbed. You can be absorbing either nitrate or ammonium or urea or organic nitrogen, which could be amino acids, proteins, soil bacteria, etc. And what we want to have happen is in each 24-hour photoperiod, when a plant absorbs nitrogen in any of these forms, we want that absorbed nitrogen to be converted into plant proteins in each single 24-hour photoperiod. In other words, we do not want the plants to experience an accumulation of nitrate or an accumulation of urea or ammonium in the plant tissue. When we are utilizing SAP analysis, the SAP analysis measures nitrate and ammonium and total nitrogen separately as 3 separate analytes. And our goal is to have non-detectable levels of nitrate, non-detectable levels of ammonium, and yet for the plant to have abundant levels of total nitrogen. That is an indicator and a signal to us that the plant is properly converting all these various forms of nitrogen into complete proteins. And when this happens, plants become very resistant to all of the insects that have relatively simple digestive systems. all the larval and sucking insects. Aphids are in this list. Leafhoppers, flea beetles are in this list as well. Spoke about flea beetles a little bit yesterday. What is needed to accomplish this? I'm missing a part of the story, I think. So in order to get to level 2 of plant health, plants need to have adequate levels of these 4 elements: magnesium, sulfur, molybdenum, and boron. So what is the pathway, and how does this process of protein synthesis confer immunity? This is an important conversation. I want to describe some of the mechanisms. I included this because of the challenges that you have with some of these insect pests. So in this process of protein synthesis, plants absorb various forms of nitrogen from the soil. We'll use nitrate as an example. Through the protein synthesis process, the initial compound that is formed is glutamate, and then that gets converted to amino acids. Amino acids get converted to peptides. Peptides to complete proteins. What this process looks like, imagine that let's imagine that you begin with this is an overly simplistic analogy, but let's imagine that you begin with a simple sugar molecule, glucose. Glucose you can visualize glucose as a short chain that has 24 links, C6H12O6. That glucose molecule is then combined with nitrogen, rearranged, reconfigured to form individual amino acids. 23 are common within plants. We have several hundred that have been identified so far. Then those amino acids are bonded together in pairs of 2 or 3 to form what we call peptides. And we refer to, you may have heard of these as dipeptides or tripeptides. These are groups of 2 or 3 amino acids. And then the next set of reactions is that these peptides are now bonded together to form much longer, more complex compounds that we now call complete proteins. There's a reason why I'm describing all of this. This is important. The process of building these compounds inside plants, for every individual reaction that occurs, every time one amino acid gets bonded to another amino acid to form a peptide, or one peptide is bonded to another peptide, for every reaction, each reaction requires a very specific enzyme. And enzymes are crystalline proteins that, these are photomicrographs of enzymes inside plants. You can do an online search for images. There are hundreds of these images online. But they are these beautiful, beautiful crystals inside plants. And I'm very intrigued and fascinated by understanding the mechanisms by which these processes work. And these are quantum and biophysics mechanisms. These enzymes are essentially, the way that I like to think about them is that they are miniature lasers. They are accumulating, capturing, and storing photonic energy, and then releasing photonic energy in a burst like a miniature laser to weld bonds together. That's a crude analogy, but that's essentially how enzymes are functioning inside plants. Now, enzymes have a resemblance to end wrenches in the sense that each enzyme can only catalyze one specific type of reaction. It can only bond amino acid B to amino acid X. That's it. It can't perform any other function. And as a result of each enzyme only being able to catalyze one specific reaction, there are tens of thousands of different types of enzymes found within plants because there are tens of thousands of different reactions that need to occur. But also, just the same as an end wrench can only fit one size nut or bolt properly. But also, like a wrench, they are not immediately used up. Each enzyme can catalyze this particular reaction over and over and over again for thousands of reactions. However, in order for these enzymes to function, each enzyme requires what is called an enzyme cofactor. Without the enzyme cofactors, the enzyme is completely dead and dysfunctional. These enzyme cofactors are all trace mineral keys, or B vitamins that are dependent on trace mineral keys. So enzyme cofactors might be molybdenum for the nitrate reductase enzyme. It might be magnesium or nickel or zinc or manganese or copper or cobalt. It's quite interesting that when you have a conversation about what are the elements that are required as essential elements for plant performance, you might get a list depending on who you ask and when you ask. You might get a list of perhaps 16 elements, or some people will give you a list of 22 essential elements. But when you ask the question, how many elements are required for plants to have a completely functioning enzyme system, you get a very different answer. It's in the 40s, at least, that we know of, perhaps more. So all of a sudden, enzyme cofactors like silver and iodine and vanadium and lithium, the list goes on, of all of these enzyme cofactors that are required for plants to have a completely functioning enzyme system. And this is the foundation of forming complete proteins, along with the other macro elements that I described. Why? Because the reason I've been describing this process is because the digestive process, whether we're talking about the digestive process of a ruminant animal like a dairy cow or a sheep, or our own digestive system, or the digestive system of an insect like flea beetles, is exactly the same as this process of building compounds inside plants, except in reverse. Instead of having enzymes to build these bonds, we now have digestive enzymes to break them apart. So we talk about proteolytic enzymes, enzymes that have the capacity to break down proteins. So again, we have enzymes that are required for each specific type of bond. We know that different animals, different mammals, different organisms have very different enzymatic digestive systems. They have very different digestive enzymes in their digestive system. For example, the contrast between our digestive system and that of a ruminant animal, we cannot extract energy from forages and from dry hay because our digestive system lacks the cellulase enzyme. We cannot break down cellulose and extract energy from it. But dairy cows can, because the rumen microorganisms contain the cellulose enzyme, and they're able to break down the cellulase enzyme and are able to break down cellulose and utilize it as an energy source. And this is the perfect analogy for the contrast between our digestive system and the digestive system of many insects. Many insects do not have the digestive enzymes that are required to break the final peptide-to-peptide bond that gets used to build complete proteins. Did you follow what I just said? Insects are incapable of digesting complete proteins. When plants have complete proteins, they are no more a viable food source for an insect population than hay is a food source for us. So this is why this whole process of protein synthesis provides immunity to all of these insects like flea beetles. It's not because the plant is all of a sudden producing immune compounds that suppress the digestive system, although that can also happen. I'll speak about that in just a bit, but that you've simply removed the food source. These plants are no longer digestible, and I want to give you a couple of quick examples. Actually, before I go into these examples, I need to tell you how, when I observed this for the first time, I haven't shared this story in quite a while. It's an incredible story. We were visiting friends in southern Ontario, and I was perhaps, I'm not sure, 14 or 15 at the time. And friends we were visiting had a family potato patch, or for several families, perhaps roughly 1/4 of an acre, at some distance from the house. And shortly after we arrived, the father said that he wanted to take us for a walk to show us his fields and show us his farm. And he said, let's go back and check out the potato field, because I walked by there 2 days ago, and the Colorado potato beetles were just beginning to emerge from winter dormancy. And there were a lot of them, and I'm kind of concerned about what the potato patch might look like. So we go for a walk, we come to the potato patch, and the soil is littered with dead adult Colorado potato beetles. And when I say it's littered, I'm talking to the order of 80 to 100. In some areas, there were as many as 120 dead adult Colorado potato beetles per square foot. The concentration was intense. And what had happened is that this potato field was right alongside an old hedgerow and that had not been well maintained, and the Colorado potato beetles were had overwintered in the hedgerow, and they were coming into the potato field from that side. These adult potato beetles were literally blown apart from the inside out. There were bits of wing and shell scattered all over the place. or simply blown apart. And I puzzled about this observation for many years until an entomologist told me, he asked me the question, he said, John, you're describing the development of complete proteins that are not able to be digested, and you also have the presence of carbohydrates. If digestion is inhibited, and instead of digestion you get fermentation, what happens when you ferment proteins and ammonia, proteins and sugars? Well, proteins and sugar fermentation is ammonium plus alcohol. And that combination does not mix. You created an internal explosion. That's pretty remarkable. We've seen that now dozens of times, and it's a ton of fun when that happens. So there are a few key pieces. One is that ideally, it is optimal to manage plant nutrition in such a way to prevent insect susceptibility and prevent these insects from ever showing up in the first place. But also, when you do have insects present in the field already, it is possible to use this strategy and to use this information, this approach, as a successful treatment for insects. That is every bit as successful. In fact, it's more successful in many cases than insecticide treatments, and also it does no harm to beneficials. So I want to give you a few examples. This is a farm that we work with in southeastern Pennsylvania a number of years ago. It's probably a decade ago or more at this point. We were just learning about this process. Can't really tell from the image all that well, but this is first year certified organic farm. They're growing 6 rows of corn and then 6 rows of soybeans. I received a call early one morning. They were quite concerned about corn rootworm pressure as well as wireworms. They had both wireworms and corn rootworm pruning the corn root system. I'm not sure which all images got included in this slide deck, but about 25% of the corn seedlings were severely stunted because of the larvae pressure, and obviously they're organically certified. They can't use an insecticide. They wanted to know what thoughts, what recommendations we might be able to make to solve the problem. We made a set of recommendations for them that included, I think, 3 or 4 AEA products that address this magnesium, sulfur, molybdenum, boron combination, along with some biostimulants and some immunolicitors. So there are several immunolicitors that we use, and an immunoliciter is the equivalent of a vaccine for a plant in that it triggers an immune reaction. It triggers an immune response. It triggers the plant to produce compounds that have these that inhibit digestion. So this product, our recommendation was applied that evening, shortly the same day that we made the recommendation. Actually, there's a modifier there. The application was made that evening, but the farmer doubled our recommendation rates. None of you ever do that, right? Good, I'm glad that's the case. So we doubled our recommendations, and yeah, I'm missing a couple of slides here, but 24 hours after the application, it took going through the soil with teaspoons to find the larvae. It was some pretty intense crop scouting, but 24 hours after the product application, the wireworm and the rootworm larvae were all dead. 24 hours after application. Now, this was with a foliar spray that was applied to the plant foliage, not to the roots. But what happened is we changed the plant's biochemistry. You changed the protein profile, you changed the carbohydrate profile, and you've removed that plant as a viable food source for the wireworm and the corn rootworm larvae. And this is now, we actually use this type of an application for treatment of corn earworm. We know that if we have plants that are not yet at a level of health where they're fully resistant to corn earworm or to European corn borer, we will wait until the corn plant begins silking, and at the green silk stage, we will put on multiple applications, as many as necessary, based on what SAP analysis indicates. We will put on applications of this combination of 4 nutrients plus these immunolicitors, and we can achieve perfect 100% corn earworm control. Because when that egg is laid and the egg hatches, and that larvae begins feeding on the silk, when that silk has complete proteins and complete carbohydrates, that larvae will never make it to the interior of the ear. This was a scenario on, actually I'm going to skip over this. This is organic corn in Kansas in 2015. I've described this example in a couple of webinars that are available online on YouTube, but do you have spider mites here? It's too moist of an environment. You don't have spider mites in England, do you? Good luck. Oh, you do? All right. So this particular farm was using sap analysis and their overhead pivot irrigation, as you can tell. The crop scout, so we had pulled a sap analysis several days prior. The sap analysis had just returned. Our consultant had made a set of recommendations for what nutrients to apply based on the sap analysis, and the application had already been started through the pivot. So instead of spraying it on, they were injecting it into the irrigation pivot. The irrigation pivot takes 48 hours to make an entire circuit. Shortly after the pivot had started, about 12 hours after the pivot had started, a local crop scout came to check out the crops, and he identified the presence of a fairly significant population of spider mites in certain sections of the field. And at this point, the application was already going on. And they came back and scouted the same sections 48 hours after the pivot had passed over them. So they waited until 48 hours after application, and all the spider mites were dead. Simply as a result of a nutritional application that was applied in the pivot. You know, it's sometimes valuable to just pause and step back and take a deep breath and get some fresh perspective. The piece that is so intriguing to me about our internal culture and the work that we do at Advancing Eco-Agriculture is that our team doesn't even get excited by stories like this anymore. This is now the expectation. This is a routine, everyday, matter-of-course occurrence that we expect to be able to control and manage insect pests with nutrition applications when they are well designed. And that organic corn crop also had no corn borer or earworm as a result. Once you optimize a soil's microbiome and its nutritional balance for optimal crop performance, then the weeds begin to do less well. And this is, I've described this in a couple of blog posts. There's some interesting research on this back in the 40s and 50s. But they describe that when you look at the Brix readings of plants, when your crop has a higher Brix reading, or in other words, is healthier, has better protein synthesis than the weeds, the insects and the diseases begin attacking the weeds rather than the crop. The weeds become the first and the most disease susceptible and insect susceptible because the soil they are less fit for purpose in that particular soil environment. So this particular lamb's quarter plant was actually on my parents' farm that I grew up on, and there was a whole patch of lamb's quarter that came from an intersection of three fields coming together. There was a tomato field, green beans, and salad greens, a combination of a mesclun mix, lettuces, spinach, a combination of different greens. Now, the tomatoes are not particularly susceptible to aphids. The green beans certainly are, and the mesclun and greens mix is highly susceptible to aphids, and yet there were no aphids on any of those crops while the lamb's quarter was being completely destroyed. Boy, that's a lot of fun. Wouldn't you like to see that happen on blackgrass for once? All right, level 3 of the plant health pyramid. Can someone give me an indication of how I'm doing on time? How much time do I have left? What's that? 15 minutes? Thank you, Kish. Goodness, where did the last hour go? All right. 25 minutes. Thank you. The third stage is when you increase lipid synthesis, when you increase the plant's concentrations of fats and oils. So I first came to an appreciation of really understanding what the importance of plant lipids when I was doing dairy nutrition. So early in the early days of my consulting career, I was also I worked closely with Gary Zimmer from Midwestern BioAg. We were working with a number of dairies. I started doing dairy nutrition for a number of 100% grass-fed dairies. Well, how do you do nutrition on 100% grass-fed dairy? You have to make sure that you balance the nutritional ratios and profiles in the forages themselves. And as I'm sure many of you are very familiar with, it is easy. There is no effort required to produce an abundance of protein. But the chronic challenge we have, particularly in intensive dairy production, is producing forages that have enough energy and supplying enough energy. And normally we think of energy in terms of carbohydrates and sugars. What's the sugar content of the crop? But as we started with this process of amplifying photosynthesis, putting on foliar applications to increase the photosynthetic levels of a plant. And yes, it's true that the sugar levels of the plant went up, but the sugar levels on the forage analysis did not correspond to the increased productivity of the forages that we were treating. And when we started sending samples to a laboratory, we quickly observed that there is this common baseline threshold. Many commercially produced crops or plants, even forages under a conventional fertilization regime, will have a minimum baseline fat content, and this is on a dry matter basis, somewhere in the neighborhood of about 1.5% to 2%, simply because that is the minimum level that is required for them to form cell membranes. When you have a dual phospholipid cell membrane, on a dry matter basis, you very seldom fall below the threshold of about 2%, occasionally down as low as 1.5%, depending on the plant species combination that you're measuring. But as we were working with these forages to increase photosynthesis, all of a sudden the fat content started increasing to, it was quite easy to achieve 4%. Even 5% was not that difficult, but 6% was a significant threshold. Once you cross 6% fat content on a dry matter basis, the animals began performing completely differently. Dry matter consumption went down, milk consumption increased, butterfat content increased. And I had this observation once. I've only had it once. I've not had the privilege of repeating it. We had one farm that produced forages at 8% fat content, and as an experiment, they offered the cows grain, free choice, when they were walking past, as they were walking through a lot to access the milking parlor, and the cows completely ignored the grain. These were Holsteins. These are Holstein cows on a 100% grass-fed diet, turning up their nose at grain. And this farm was a blended operation of both organic. It was an certified organic, raw milk, 100% grass-fed dairy in Lancaster County, Pennsylvania, and they also had about a 30-acre vegetable operation. And at the end of that year, they decided that one of those two has to go because they can't continue to do everything, and they sold the cows. And I was so disappointed because I wanted to see that again the following year and repeat that experiment. So what happens? This was the initial learning and trigger. But then I started asking, one of the things that we observed and noticed is when you have plants that have surplus levels of energy, when they have these abundant increased lipid synthesis, you can actually see this visually in the leaf or on the leaf when you have this glossy, waxy sheen on the leaf surface. And that glossy, waxy sheen, this wax expression, is an expression of abundant lipid synthesis. So what does it take to get here? In order to get to level 3 of plant health, plants must absorb the majority of their nutrition in the form of living microbes or microbial metabolites. As I described yesterday, I described the rhizophagy cycle and rhizophagy process. I'm not going to go into it right now. I'll touch on it again in my next presentation. But you will not get to level 3 of plant health as long as plants are absorbing the majority of their nutrients in the form of simple ionic fertilizers. It's simply not going to happen. And here's an overly simplistic explanation of why. When I'm going to use nitrogen as an example, and I'll expand on this topic also in my next presentation, but what happens is, now, let's say that you want to build a house, or even better yet, you want to build a tractor, and you have two choices. The first choice is that you can purchase all of the pre-manufactured parts, or the second choice is that you have to mill and machine all the parts individually one at a time, and then assemble the tractor. Which is going to take you the least amount of energy? Combining the prefabricated parts, of course, takes just a fraction of energy as it would if you had to build all of your components from their foundation. And this same concept also holds true of plants. So when plants want to build complete proteins, I'm going to use proteins as an example here, as a reflection on nitrate nitrogen. I'm innocent. I'll go with the two extreme opposite ends of the spectrum. On one end of the spectrum, plants have the ability to absorb microbial cells and utilize proteins from those microbial cells and incorporate those proteins directly into their cells. So it is the equivalent of getting prefabricated compounds. The microbes have already done all the work. The microbes have pre-assembled the proteins, so this whole protein synthesis process of amino acids, peptides, proteins, etc., doesn't need to take place inside the plant cell. In other words, the plant needs to spend no energy. There's no energy spent in utilizing microbial proteins. On the other end of the spectrum, you have plants absorbing nitrates, which is the most common form of nitrogen that is absorbed when chemistry fertilizer is applied. When a plant absorbs nitrate, it requires two things in large quantities to convert nitrate to proteins. One is it requires a lot more water. For every molecule of nitrate that the plant absorbs, it requires 3 molecules of water to convert to glutamate. And the second thing it requires is a great deal of sugar energy, photosynthetic energy. When a plant absorbs nitrate, actually this particular data point is on corn. It's a plant that's very well researched, but this holds true for many other plant species, or for all other plant species, I should say. When a corn plant absorbs 80% of its total nitrogen requirement in the form of nitrate, it requires 16% of its total photosynthetic energy, its total sugar energy, just to convert nitrate to glutamate and to complete proteins. That is a tremendous energy drain on the plant. So providing a plant nitrogen fertilizer in the form of nitrate actually has the ability to produce a yield drag effect because it reduces overall plant energy. And the way that this manifests and expresses itself is because it reduces lipid synthesis and it reduces fat content. So I need to keep moving here, but what happens is that when plants get to level 3 of plant health pyramid and have these abundant levels of lipids, they, at this point, this waxy shield on the leaf surface, this waxy cuticle layer, confers resistance to many of the airborne fungal and bacterial pathogens, so rusts and mildews, etc., bacterial diseases. What happens is that all of these pathogens land on the leaf surface, and the great majority of them release enzymes that are termed pectolytic enzymes. In other words, they break down pectins. And when they land on a leaf surface that has the thick layer of waxes and lipids on it, then this is a very effective shield that never allows the pectolytic enzymes to get access to the pectins in the cell membrane in the first place. And you prevented an infection from ever showing up. So I've already described this. Plants require a very aggressive microbiome in the rhizosphere in order to achieve this level of plant health. Then we get to level 4. Now, actually, it's been a while since I put this slide deck together. Well, we'll come to this. So at level 4, plants begin developing increased concentrations of these plant immune compounds, phenolic compounds, terpenoids, bioflavonoids, compounds that we in plain English can simply call essential oils. There's tens of thousands of these different essential oils. All plants produce these compounds as plant protectants to protect themselves from ultraviolet radiation, from overgrazing, from insect attack, from disease attack. Many of these compounds have very potent antiviral, antimicrobial, antifungal, antibacterial properties. And their mechanism, the way you mentioned, you may have missed or noticed a moment ago, I said that they're there to protect themselves from overgrazing and from insect attack. Many of these compounds also directly inhibit digestion. So this is another mechanism, another layer of mechanisms for messing with the digestive system of insects, is that there are some of these compounds that are produced directly inhibit digestion in insect digestive systems. So to get to level 4 of plant health, what happens is that the plant immune systems, there are two immune pathways. The SAR pathway, which stands for systemic acquired resistance, ISR stands for induced systemic resistance, and they're both triggered by microbes. These two pathways are triggered by microbes in the microbiome, both inside the plant, in the rhizosphere, and the phytosphere. And at this stage, plants become resistant to the entire beetle family: Japanese beetles, Colorado potato beetles, marmorated stink bug, corn rootworm beetle, and flea beetles. Although flea beetles can also happen at level 2. And we also get resistance to nematodes at this point, as well as viruses. Plants require the correct microbes in the microbiome to produce this outcome. So there are two very distinct groups. If you noticed, if you were following along and paying close attention, you noticed that level 1 and level 2 are purely a function of chemistry balance. This is nutritional balance. You can grow plants that are at level 1 and level 2 of plant health in a hydroponic operation. You can do it hydroponically. But not so with level 3 and level 4, because level 3 and 4 are a reflection of the microbiome. They are a reflection of biology that plant is associated with. And also, the lower two levels, level 1 and level 2, are what I describe as passive immunity, where we simply remove the food source. We change the plant's biochemistry profile to such a degree that they are no longer a viable food source for the diseases or insects. And then at level 3 and 4, this changes, particularly at level 4, where we switch from passive immunity to active immunity, where these plants are actively producing compounds that, quite frankly, simply put, have the ability to kill. These essential oils have the ability to kill. They are the plant's self-protection warfare mechanisms. They have the capacity to kill bacteria, to kill fungal infections, and to also kill insects that would desire to feed on that plant. All right, I think that's a wrap. I shouldn't be advertising the book anymore because I think we were down to under a dozen copies by the time this morning's book signing was over. And you can find more of our information here and find our blog here. So thank you for that, and we're going to open it up to Q&A. Thank you. Thank you. Yes, we have, I'm not sure where the microphone is, but we have a couple of questions here up front, okay? Microphone is coming. I have a lot of fun with these conversations. I particularly have a lot of fun describing them to people who haven't heard them before, because there is so much opportunity. And one of the things that I'm quite excited about when one of the farm visits that I was on yesterday or on Tuesday is that many of these pieces are so easy to implement and to execute. You have such a golden opportunity to make a significant difference very quickly. Yes.
Hi, John. Thanks for that. It was brilliant. Just in regards to the comment you were saying about calcareous soils, you mentioned magnesium, manganese, and iron. In what form should you be applying those?
In the case of manganese and iron, magnesium is relatively easy and straightforward. There are different forms of magnesium that you can use. I would suggest you consider using magnesium sulfate as one that's readily available and affordable and easy to apply and produces a consistently nice response. For manganese and iron, there are two particular considerations. They need to be in the reduced oxidation state, and they need to be chelated, and they need to be applied as a foliar. At least in the short term, as you are in the process of regenerating soil biology to the point where it has the ability to produce these reduced forms of manganese and iron in the soil profile, they need to be applied as a foliar. Because what will happen otherwise is that you purchase reduced and chelated manganese and iron and you apply them to the soil, and over a period of time, they revert right back to the oxidized state because you have such a severely oxidized soil environment. So the most direct and least expensive and most effective pathway is to apply those as a foliar. That's where you're going to get the greatest crop response. Yep. We had another question, yes. Oh, over there.
Hi, yeah. You mentioned that it takes tens of thousands of enzymes to produce the proteins, but there's 23, I think you said, amino acids. So combining 23 amino acids with another 23 amino acids means you only need 529 enzymes. all bacteria produce the same amino acids. Their genome size are so small, they can't have these tens of thousands of combinations. They only have, 3,000 maximum number of genes, potentially 1,000. So how does that relate to what you were saying? I agree, secondary metabolites, very important, useful for antifungal, antimicrobial, anti-insect feedants, but I don't like the logic that you're applying.
Which logic specifically? I was trying to follow you there, but I didn't quite succeed.
So you're talking about the number of enzymes that it
Yep.
requires to produce all these proteins. If there's only 23 amino acids, each of those amino acids can combine with 23 other amino acids. So
Yep.
you actually only need 529
Yep.
enzymes, but in fact, quite a few of these enzymes can do multiple combinations.
Yeah, so 2 points. First of all, this is not my domain of expertise, and I could very well be mistaken and be misinformed, and I would be happy to be corrected. It's my understanding that there are enzymes not just to bond amino acids, but also to peptides, and also that while there are 23 common amino acids, there are close to 200 in total that have been identified altogether. But also regarding the number of enzymes, this is not a number that I derived mathematically or came up with. It is direct reference in the literature, and I'd have to find the citation for that. But it's my understanding that the number is much larger than just in the hundreds. Yeah. All right. Well, more homework.
Hello.
Yes.
Just interested, really. So, how do you protect a... Hello. That's better. How do you protect a oilseed rape plant that just comes through the ground against pests if you haven't got the leaf area to apply nutrients through?
Yes. Outstanding question. Thank you. We also have questions up here in front and then more on the back. Very good question. And the answer is you need to address those nutrients as a seed treatment. And we've experimented with this extensively. We actually launched a new product this spring that's called Seed Flare that is a mineral seed treatment. And we are taking these trace minerals that are in the reduced form and chelated and applying them as a seed treatment, as a liquid seed treatment, at very concentrated dosages. So we are applying a total liquid solution, varying on the seed, moisture content, seed size, and so forth. We're applying anywhere from 8 to 20 liters per ton of liquid. And what happens is within about 24 to 48 hours after treatment, all of that liquid is actually absorbed into the seed. The seed still appears to be completely dry on the surface. And you now get this immune response or this resistance response that we are looking for immediately upon germination. Yep. Yes.
Hello. After listening to your recent podcast where Christine Jones was saying that legumes produce nitrates through their nodules, do you feel that if we're growing too many legumes in our systems, we're at risk of never being able to get to level 2 on the health pyramid?
The direct answer is no, but there might be some misunderstanding in the way you're asking the question. So first, it's not that legumes don't directly generate nitrate levels in the soil profile, because in their nitrogen fixation process, the metabolites of that are both organic nitrogen, and that eventually some of it may convert to ammonium. But then through the nitrification process, as rhizobiums slough off or the soil microbial community cycles them, then you may end up with some nitrates being generated from legumes. So there is an indirect process in that. Yes.
Hi, we use a lot of companion plants like tagetes against root-feeding nematodes, and I heard you talking about fenugreek for flea beetle. Would it be worth making extracts of those and applying them as a foliar? Do you think that would also be beneficial?
I don't know. It's worth experimenting with, but yeah, I don't know. Any other questions? Oh, quite a few here in the front. Let me see. Okay.
John, talking of foliar applications now, how important is the water source, pure water as opposed to tap water?
The water source for foliar applications is extremely important. In fact, it's so important that we produced an entire webinar on the topic that is called Creating a Well-Designed Foliar Application. It's about an hour-long webinar. It's available on YouTube. I highly recommend it. The short version is that there are several minerals in water that can really impact the effectiveness of a foliar application, but bicarbonates in particular, and it requires a surprisingly low level. When you have total water hardness that is as low as 150 parts per million, that can reduce the effectiveness of materials that you add to the spray tank by as much as 70%. It's a significant reduction. So if you want to improve the effectiveness of foliar applications, making sure you start with high quality pure water is the foundational starting point. Test it and measure it and know what it is and where it needs to be. It's not that expensive. Yes?
Hey, thanks for the great talk. So as a side remark, the moth you are showing, well, it was having its lovely feathery antennas to have sex, not to find the plant, but that's a side mark. But the real question is, what about the plant-insect chemical communication? It has been very well established that this is the main way that plants and insects actually communicate. And the fact that insects are not stupid, they like to have healthy plants, they want to have good nutrition from the plants, and they can read it from the chemistry and the overall how the plant is looking. So they don't want to have shitty food. They want to have the best nutrients in the plant as well.
Exactly my premise. But what is ideal, an ideal food source for an insect or for a moth is not an ideal food source for humans. If you want to dig deeper into insect olfaction and insect communication systems in the environment, I would suggest that you follow up one of Philip Callahan's protégés, the last PhD student that he supported was an entomologist named Dr. Tom Dijkstra. And I've interviewed Dijkstra on the podcast. He's conducted a couple of webinars where he goes into some detail on the challenges with insect olfaction and the foundation for insect electromagnetic communication systems. So there's something for you to look at. Yes.
Hi. Yeah. So having worked a bit with SAP analysis, I've noticed value systems are not apparent for every kind of crop.
Yep.
So I wonder if you have any protocol to produce that value system for yourself. Is there any way to actually manage that?
Yes, we do. I'm not about to give you the magic sauce here in front of everyone, though. Yes, so in concept, you need a reasonable volume of samples from crops that are known to be performing well. So from crops that are actually resistant to diseases and insects, that are actually producing high quality harvests and abundant yields. And when you have a data set of crops with those characteristics, it's relatively straightforward to compare their nutritional profiles with the rest of the data set and evaluate where the nutrient ratios need to be for optimal performance. Yeah.
John, some people are experimenting with their own seed dressings, the likes of lactobacillus or even a vermicompost, on the seed themselves. Maybe not directly the nutrition you're talking about for insects, because insects wouldn't be the biggest burden we have where we're from. Do you think those are beneficial as biology on the seed instead of traditional seed dressings, which we're trying to get away from?
Generally, broadly speaking, the answer is yes. Obviously, the actual performance can vary widely based on the product itself. But in general, I'm a big advocate of adding biological seed treatments and mineral seed treatments to the seed as well. So yes, those tend to perform very well. Yeah.
I'm an organic farmer from the Netherlands, and I was wondering about the weed topic that you were gonna maybe address. But do you have some key takeaways for me in that matter?
From a foundational principles perspective, plants thrive in soil conditions and in different succession stages as a reflection of three foundational characteristics of the soil: as a reflection of the soil microbial community, as a reflection of the soil mineral balance, and as a reflection of soil physical characteristics. And this is from a broad overarching perspective. When you have soils that have good aggregation and good aggregate stability to a significant depth, preferably below the A horizon, and you have abundant microbial communities and balanced nutritional profiles, balanced calcium to magnesium ratios in particular, and avoiding excessive phosphorus and potassium and nitrogen applications, that set of conditions usually leads to scenarios where the crop plants are healthier than the weeds, and the weeds become the susceptible hosts for diseases and insects. Yeah. All right, one more question? Yes.
Thank you. Quick one, just thinking about, slugs and snails, so mollusks, are their digestive systems similar to the lower insects that you're talking about, and would level 2 confer resistance to them?
There's actually a different mechanism. I've been, for the last 2 days, trying to think of the name for the fluid for their circulatory system, and it's escaping me. I need to look it up. But instead of having an iron-based hemoglobin, their oxygen transport compounds are copper-based, which means that fluid is green instead of red. And this is why they have a particular susceptibility to high levels of iron. So when you look at all the commercially available slug baits, are all some form of iron that is put into an attractive form to be used as a food bait for slugs. And our experience has been that there are, well, I'm not going to go into the, there's quite a bit of science behind plant wilting expression that enables or increases susceptibility to slugs. But our experience has been that when we foliar feed iron and make sure the plant has generous levels of iron, slug problems just tend to disappear. All right, thank you very much.